In this third of a series of three on this lecture A of cardiovascular physiology, so we are calling it Cardiovascular Physiology 1, this would be dash 3. We are going to consider the cardiac myocyte, some of the anatomy again, to expand a bit on what we had seen before, and now place it in the context of excitation contraction coupling that is different in the cardiac myocyte than in the skeletal myocyte. And then we'll proceed to consider the action potential and how that is seen, or we'll see it anyway, as it occurs in contractile cells. So quite obviously, since we're emphasizing contractile cells, we know that there are pacemaker cells in the heart and conduction fibers as well. So we hadn't shown that with the anatomy of the heart, but certainly it is expected that you recall that there are like the Purkinje cells that are modified cardiac myocytes that are the conduction fibers to conduct the event, the pacing event from the pacemaker cells at the sinoatrial node to the AV node, and then from the AV node down into the ventricles. So I'm expecting that you can picture that as I'm describing it. So with the physiology, we're going to look first at the contractile cell, the target cell of the conducting fibers. Then we'll talk about pacemakers and possibly the conducting fibers as well. But the contractile and the pacemakers are the important ones. So now we're considering just the contractile cardiac myocytes. So because it's the contractile ones, we can safely make a comparison on that level with the skeletal muscles. So overall, as you recall, they're smaller. They get they are mononucleated, and they have branching cytoplasmic processes that interconnect with the branching cytoplasmic processes of other cells, cardiac myocytes. The contractile ones we're thinking about in the wall of the ventricles. Or the auricle of the atria, and they have the intercalated discs, as you recall, with the desmosomes that allow the force to be transferred, and it's the gap junctions within them that we had mentioned before in thinking of the conduction or the transmittance of the depolarizing wave. So the desmosomes are equally important because the force dispensation from one to the next. The T-tubules are larger and branched, and the cytoplasmic reticulum is smaller, sarcoplasmic reticulum, and the mitochondria occupy 1/3 of the cell volume. So obviously the oxidative phosphorylation, the ATP generation that is required in a cardiac myocyte is extremely higher because it never stops. If it does, you do. You can rest the skeletal muscle, and then it's just got the tonic activity that can alternate from motor unit to maintain tone and allow neurons and skeletal myocytes time to sustain a homeostatic baseline level of energy within the skeletal muscle cells in anticipation of a demand. Well, there's a constant demand on the cardiac myocytes, obviously. So the intercalated discs that we see here, they are scattered with the desmosomes and the gap junctions. But also, if we look down below here at this one, there is the interdigitation of those. So there's an inward projecting part here. I guess we can see it there. One end will go in, and on the other surface they will project out. So this obviously gives more surface area for the attachments, the series attachments. Right, this is series, one end to the other. The series of attachments to dissipate the force, as well as to increase the surface area for the ionic flow to send the depolarizing wave. I was just looking quickly to see if they showed the T-tubules and things like that, but we'll have to recall that anatomy from the previous lecture, and that the action potentials will be generated on the surface of these cardiac myocytes. You can see the myofilaments within the cardiac myocyte. It ends there within that particular one, but it continues to the next one. And so we are envisioning the sarcoplasmic reticulum in here, along with some T-tubules, and so the action potential is going to go along the sarcolemma, the cytoplasm of the cardiac myocyte, and it is going to go down through the T-tubules and be associated with triggering the release of the calcium for the contribution to the cross bridge linkages to occur as we had described before. Now what we want to do before leaving this slide is just note the orientation of the cardiac myofibers in series as they swirl around the ventricles. This one is nice because it reminds us that when we look on the anterior surface of the heart, we should see predominantly the right ventricle. The problem I'm having with it is that they're showing the muscle tissue, the cardiac muscle tissue of the right ventricle becoming continuous with the left ventricle that we know we can see over here. They actually will dip in deep here to the interventricular septum. So I'm not pleased with this illustration. But what it does show is that there's a spiraling type of a relationship of these. So it's not a linear contraction event from the tip of the ventricle toward the output. It's more of a coiling. So you can picture your two hands with some of your fingers interdigitating around a balloon, and you kind of twist your hands to compress that balloon, and then back and forth to release, to fill again, and then contract. So it's not just a lengthwise, like your biceps pulling your forearm close to your shoulder to pump the blood out. It is a true compression, a spiraling type of a compression. So now we're going to zoom in just to a single T-tubule where it is again going to be associated with sarcoplasmic reticulum. We're looking at the actin and myosin, the thick and the thin filaments, being nearby with sarcomeres that look very much like, and indeed they are in essence identical to what we would find in a skeletal muscle cell. So I say in essence because it is entirely possible that there are some associated proteins that would differ in the cardiac tissue versus skeletal tissue. But when it comes down to it, the basic myosin-actin interaction being directly dependent upon two components, the calcium concentration, the availability of calcium, if you want to word it that way, and the ATP concentration. Remember we said all the mitochondria, or in other words, the availability of ATP. Those are the two key dependent variables. Excuse me. They would, yeah. Anyway, they are actually the independent variables. If you're doing an experiment, you would change those two to see their effect on this. But there is a direct dependency of the contractile event on those two variables. So we're going to follow through in the series and see how they differ. And what we remind ourselves before we get started, we're going to follow it in a series so we can make sense of it. But all these processes are ongoing at any instant in time constantly in the cardiac muscle tissue because it's constantly beating. The heart is constantly beating. It's constantly contracting and relaxing. So these are ongoing all the time. So these are enzymatic or transport protein steps, or both, the ATPase plus the transporting that's occurring here. They're ongoing all the time. The opening and closing of channels is not just open, close, but open, close, open, close, open, close, open, close. So ongoing cycling events all the time as well. So in other words, the efforts to reverse a contractile event are equally ongoing as the efforts to cause a contraction, but there is a temporal triggering of the action potential and properties of the membrane to create a very interesting relationship between the duration of the action potential and the duration of the contractile event in a cardiac myocyte, and that'll be the last part of this presentation we're considering now. So again, it's the ongoing events, the gas pedal and the brake story all the time to produce a homeostatic baseline level of our two key players, ATP and calcium, in the cardiac myocyte. Now, what they're showing here as well, before we start going through it numerically, is the state of contraction, the shortest sarcomere on the left, and the lengthened sarcomere on the right. So I challenge you to think of the anatomy of the left ventricle. We had talked about initial relaxation immediately after emptying, and then you would begin with the relaxation and the dilation of the ventricle. So what we're seeing is the progression from this point where calcium has been removed to a certain level to inhibit the cross-linking, and then you can begin to have the lengthening. Again, we got to think of these continual processes. Beats per minute. 80 beats per minute is a typical resting heart rate. It can get up over 100 beats per minute. These things are happening very fast all the time. So it's a continuum from this end state to this beginning state and everything in between. So we reduce them to a series of staged steps and make it appear as if nothing else is going on in the cell while this action potential is coming in. We know that that isn't true, but we do it this way for simplicity, and the reduced model helps us to understand the different steps that are simultaneously occurring at any one point in time. And that is, the term simultaneous, as we said, applies to everything except when we get to the context of how long is our action potential and how long is our contractile event. It turns out in cardiac muscle tissue they're the same length, so we have to understand the properties of the steps that we're talking about here and the properties of the membrane for generating the action potential, such that we get an equal time period of the action potential on a cardiac muscle cell in its entirety related to the series of events we talk about that happen with that action potential to get a complete contraction relaxation event within the same time point that we have the action potential starting and ending. Okay, so back to simplicity. One action potential gets down the T-tubule membrane to the L-type calcium channel. So this is a bit different than what we had talked about with the skeletal muscle tissue. So we're already seeing at least one membrane protein different in cardiac versus skeletal muscle tissue. And note that we're saying the action potential enters from an adjacent cell. We'll get autorhythmicity a bit later, but every cardiac muscle cell membrane has leaky sodium channels, is what they are, and they will produce the transient type of membrane as we saw with the smooth muscle. So every cardiac muscle cell, even the contractile ones, have a capability for autorhythmicity, but because we've got the pacemaker cells, we send a signal to it at a rate higher than the intrinsic rhythm, so we are sending an action potential from adjacent cells that sets the pace of the entire heartbeat. So it's important to put that in place now, because we'll start talking about autorhythmicity, and you might say, well, that slide back there said that it's coming from an adjacent cell. So it's coming faster than what the cell could make itself, so the cell is told how fast to go. If the signal coming from the SA node ever falls below the autorhythmicity of this cell, it's going to start beating at its own rate and ignore the too slow of a pace coming from the pacemaker cells. So again, another pathology brought in here. If somebody has an extreme beta bradycardia, very low heart rate, then you can start having the ventricle cells create their own pace, and you end up with an ectopic pacemaker, and you can get premature ventricular contractions. So it's really important to understand that at this point as we're playing with these drastically reduced models. So back to the simplicity. The AP, action potential, gets to the L-type calcium channel. It is voltage dependent and opens. Extracellular calcium enters near the sarcoplasmic reticulum and through this voltage-gated L-type calcium channel. And the localized calcium concentration that's generated there induces a calcium release through the ryanodine receptor channels. So we still have the ryanodine receptor channels present, but now they are triggered by calcium coming in. So rather than in the skeletal muscle, we talked about flushing a toilet, where you push one lever here and then it pulled something out there to open these. Now we're talking about a chemical signal, the extracellular calcium entering the cell. So the calcium influx creates a local increased calcium concentration, similar to vesicular release that we talked about, exocytosis. And the calcium-sensitive ryanodine calcium channels on the sarcoplasmic reticulum open, and then calcium stores, the intracellular calcium stores, will be utilized to cause an increased intracellular calcium. So I want to go back and reword that just a little bit. They don't increase the intracellular calcium concentration. That's one way to think about it, but what they do is they contribute another burst of calcium to the local calcium concentration or calcium pool, and then that can diffuse away to contribute to the overall cell intracellular calcium concentration. Remember, the strength of the contraction is going to be correlated most directly with the intracellular, the complete intracellular calcium concentration. We're just looking at one T-tubule, one little piece of sarcoplasmic reticulum. The other thing that I don't like about this is they're showing this little piece of sarcoplasmic reticulum there on the left of this T-tubule, and this one over here on the right, and they're going to show the reuptake here, obviously. Well, I tell you, this calcium molecule, if he happens to bump into a reuptake pump right there, he goes right straight back in, because it is the law of mass action. Wherever this happens to diffuse to is random, and whatever it bumps into, if it bumps into the troponin, then it'll and the actual calcium binding site on the troponin, then it's going to lead to the events that we know follow that. If it just bumps into nothing and ultimately ends up randomly bumping into the reuptake pump right here next to where it came out, it goes right back in. So keep that in mind at all times. So, big distinction to compare and contrast. First contrast of skeletal to cardiac is that we have a voltage-gated calcium channel in the T-tubule, other than the anatomical difference of the T-tubule, and the calcium diffuses in and mediates the opening of the ryanodine receptor calcium channels in the sarcoplasmic reticulum, and the intracellular calcium store is used to contribute to the overall intracellular calcium concentration. I'm calling it a calcium spark or spike, and that summed calcium sparks create a total calcium signal. So that's what I'm saying, is that we have to think of all the sarcoplasmic reticulum in all of the cardiac myocyte, action potentials spread in all directions along the membrane. So there is a total cellular spark distributed throughout the cell where all these events are occurring, and they all collectively contribute to what we might be able to physically measure or mathematically consider as the total intracellular calcium concentration, or free calcium concentration, which is the calcium signal. So that summed calcium spark making a signal contributes globally to the ability for the myosin-actin interaction and leading to the contraction. So you note that we're not going to the level of detail as we did in skeletal muscle because you should already know it. If you can't recall it all, then you should go and review it, because it is fair game for any type of assessment of your understanding of what actually contributes to the strength of contraction within a cardiac myocyte. And then to bring about relaxation, there will have to be something that will be reducing the intracellular calcium concentration such that it would favor calcium leaving troponin versus staying close and more frequently bound to it. So remember the law of law of mass action. Apologize for that misstatement there. Applies to the dynamic interrelationship of the binding site and the ligand that will bind to that binding site. And the way that we unbind the calcium is we have to have mechanisms that they place numerically after this step that will effectively reduce the total intracellular calcium concentration and favor calcium in the free form more so than being bound to the, I should be pointing to the red, the troponin on the actin filament. And the reason I go into explaining it is you can memorize these steps and then you end up with an exam question that talks about how you have to reduce the concentration. You know, steps 8 onward have to occur really to a significant degree. It's all occurring simultaneously to a significant degree to reverse this episodic event that put more calcium in. So within a period of time, that will be removed simultaneously, and the contractile event that you created is reversed. And so step 8 is the ATP-dependent calcium pump within the sarcoplasmic reticulum. So we're seeing, again, our two primary factors upon which the contraction depends, the calcium and the ATP. The contraction relaxation event, the ATP for the myosin ATPase, the calcium for the troponin binding, and the ATP we see first here for the calcium, ATP-dependent calcium pump at the sarcoplasmic reticulum. There is a second calcium pump, and this is to efflux out of the cell, the cardiac myocyte, and this is a sodium calcium antiporter, the NCX transporter. So the NC, N is the natrium, C is the calcium, and X is for exchanger. But these are called antiporter membrane pumps. If you go back to your review lectures, you remember there's symporters and antiporters. This is an antiporter. So obviously extracellular sodium is going to be at a higher concentration. The influx, even with this type of a transport protein, is going to be favored. 3 will come in for every calcium that will exit. So you should start thinking, well, as soon as we put calcium into the cytoplasm, it creates more calcium available to drive the calcium out and favor sodium coming in. If we have sodium coming in toward the end of the contractile event, we're getting ready to trigger another action potential. We're starting to depolarize already. Okay, so these are important things to recognize. When sodium comes in, you can contribute to a depolarization of the membrane, so always keep that in mind. Simultaneously, however, you have the sodium-potassium ATPase right nearby to pump that sodium out, 3 out, for every 2 potassium in. So it would almost seem like this would be negating the events. And if we are thinking about the end of the action potential at the same time that we're getting the end of the contraction relaxation cycle, then the sodium-potassium ATPase pump should be just kind of winding up its reversal of the potassium event. So remember your action potential of when does potassium efflux contribute and when is it really important for the sodium-potassium ATPase pump to be pumping the potassium back into the cell. That's toward the end of the refractory period when you're going to be reaching a resting membrane potential. It's transient in these cells because it never rests, right? You don't talk about a resting membrane potential in a cell that never rests. So there's a transient membrane potential sub-threshold. So this dynamic process, picture what happens. You just had added a bunch of potassium to the outside here, and you are bringing that potassium in. At the same time, you're pumping out sodium that came in with the action potential, as well as now you're getting a little more sodium coming in because you have more calcium pushing out on this. You can think of it that way. So drawing in a little more sodium. So you're beginning to get a little bit of a collection and accumulation of sodium just inside the membrane at this point. So it's a very complex process, and it's all happening simultaneously. So picture the initiation of the action potential. Actually, what you need to do is to take all this and superimpose those same systems right here. So just as soon as this action potential has ended here, you have, or at the end of it, you have the potassium being reversed, and right, and the sodium also being reversed. Just as we talk about the sodium-potassium ATPase pump being very important for reversing the sodium influx and the potassium efflux to bring the potassium back in and put the sodium out. Right over here, that's occurring. At the same time, the action potential just caused an increase in intracellular calcium, and that intracellular calcium is going to be pushing out and pulling in new sodium and beginning to depolarize this membrane as soon as it can be depolarized. So we're beginning to see some of the properties, the unique properties of the cardiac myocytes that give them an autorhythmicity. So the cardiac muscle contraction can be graded. The sarcomere length affects the force of contraction, and the action potentials vary according to the cell type. So what we're going to look at first is the action potential of the myocardial contractile cell, because the first thing that we want to talk about, and what we end with this presentation before we switch over to the second half of the slide sets for this particular chapter, this topic of cardiovascular physiology, is to look at how the action potential relates to the excitation contraction coupling event, or the contractile event that we just discussed. And again, you've got to go from these reduced pictures of just a little piece of the cell to the whole cell. So sometimes the authors or the slides don't take you from that level where you zoomed way in and you're standing, you're only as big as an action potential on a little piece of the membrane, looking at the neighborhood, versus stepping back and being a giant and looking at the entire cell. So now we're thinking about measuring an action potential event from a single cardiac myocyte. What is the time scale of that? And then we'll superimpose it upon the same time scale to look at the development of tension in that cardiac myocyte. But we also know that we could, if we had the sophisticated equipment, we could go and look at just a piece of the membrane and see the same action potential right there, and ask questions about what's happening for that action potential on that little patch of membrane. But now we're backing off and saying, let's think of an action potential just being instantaneously present on the entire surface of a myocardial contractile cell, and then within the cell, the collective events, the summated events of all the T-tubules, all the sarcoplasmic reticulum, and all the transport proteins and channels that we had just considered in one little piece of the cell in the previous slide. So did you reassemble your full cardiac myocyte yet? When you have, let's proceed. So we're measuring a single cardiac contractile cell. And we're, let's think of left ventricle. Let's stick with that thing that we thought about. Sometimes that's the easiest one to work with, and then we can project from there to the right ventricle, and then we can project from there to the atria and see how they might be the same, the right and the left, but different certainly than the ventricles. So the membrane potential is shown on the y-axis, and we see minus 100 and minus 80 millivolts. And so the first thing that jumps into our mind is this is a resting or a phase of the transient membrane potential at its lowest level, the trough, that we might think of as a resting membrane potential if this cell was ever going to rest, but it never rests. So the lowest, the most hyperpolarized point of the transient membrane potential is minus 90. You very likely will read in textbooks that the resting membrane potential of cardiac myocytes is minus 90. And yes, I'm recording this one right along with the previous one, so you hear the dogs. They're now back from shopping. Okay, so the first event that's going to occur is an increase in the permeability of sodium. So there are specialized membrane channels that leak sodium, and this contributes to that transient membrane potential. So as the permeability of sodium increases, remember your Goldman equation, we're going to add calcium to our Goldman equation in this particular case for the cardiac myocytes. So we have sodium, potassium, and calcium as key players now. So we're going to see the sodium permeability changing, And as it does, we get a sodium influx. The sodium influx is going to make the inside of the membrane more positive, so we get a deflection toward the positive side, right? And if we take it to the Nernst equation for the equilibrium potential, as you have one ion change its concentration across the membrane, notice I said just across the membrane, the potential of that across that membrane will approach the equilibrium potential for that particular ion species, and it is positive, as you recall, for sodium. So we are first going to see the phase 0 of the action potential of the contractile myocardial cell, where we get a transition from the minus 90 to about minus 40. And certainly you should recall that minus 40 is relevant to the threshold. And then the phase 2, we're going to see those voltage-gated calciums closed by the time we reach the peak of the action potential at about positive 20. So you also note that the peak of the action potential is different in the peak value of the action potential is different in the cardiac myocyte than the neurons. The question I pose to you is the potential difference across phase 0 to phase 1 equivalent to or different from neuronal action potential and skeletal muscle action potential. Okay, so you can look back and just subtract the highest from the lowest, or get the difference. Is that difference the same as the difference you get here? The peak value plus 20 is obviously not as high, but it is still closer to the equilibrium potential of sodium. So again, what we have is we have leaky channels that are going to get us to a threshold voltage that is going to open the voltage-gated sodium channels, and then by the time of we reach the peak millivolts value of the action potential, those sodium channels will have closed. So it's the same thing. Across the membrane, you've got populations opened and closing and closed, or inactivated, and then they still have to get reset. So we do have the absolute refractory period. And then as the sodium influx begins to taper off, we get a slight shift back toward the negative side of things, right? So a drop back towards 0, and then it begins to the rate of decrease begins to reduce, right? So here we have a rate of reduction in the membrane potential towards 0, and then that rate begins to disappear and plateau. So that rate reduces until ultimately we have almost a horizontal line, where the slope of a horizontal line is what? 0, right? And so we call that a plateau. Well, what's happening here is we have a low, a decreased potassium permeability. The fast potassium channels are closing. So there's a fast potassium channel population that's going to contribute to some of this repolarizing event, but they will close. And then there is a calcium channel that is going to open, and as the calcium concentration inside the membrane increases, or as the calcium accumulates there, then the calcium-dependent potassium ion channels are going to open. So there's two different populations of potassium channels in the membrane of the cardiac myocyte, the sarcolemma. but there's also a calcium ion channel. So remember we said we're going to be talking about permeability of sodium, potassium, and calcium. But because we have different populations of actually sodium and potassium ion channels, there are different phases where the permeability for those ions will change at different rates. And I say change at different rates because this is an ongoing process. You can look at any one point in time and say, what is the permeability at this point? But when we're looking at a segment over a period of time, it's the rate of change of the permeability over that period of time. So try to make these dynamic processes in your head. But we can make them as stop points. At stop point 0, the end of phase naught, when it's a 0, they say it's naught. So phase naught, at the end of that, you have a transition from the leaky sodium channels to the voltage-gated sodium channels. They open at the end of phase 1. Those voltage-gated sodium channels will have closed, and you know that they need time to reset as well to become capable of being activated again, opened again. And then there's some potassium, and that potassium, early potassium permeability ends at a point where calcium kicks in. So this plateau is actually due to calcium. So the increased calcium permeability, increased calcium influx that accompanies the action potential, the L-type calciums channels, as you recall, is what will extend the duration of the action potential in a cardiac myocyte. So these L-type calcium channels in the membrane of the cardiac myocyte are extremely important for extending the duration of the action potential such that it matches the excitation, contraction, relaxation event. Well, as so the negative feedback for this calcium plateau is the calcium-sensitive potassium channels. As the local calcium concentration increases, it's going to open a subset of calcium-dependent potassium channels, and you get the potassium efflux, which, as you know, will contribute to the repolarization phase. And then the phase 4 is the repolarization to resting potential, or it's not resting potential, it's the lowest value of the transient membrane potential. And what has to happen with the refractory period is you've got to have a sinoatrial node pacemaker signal get to this before its intrinsic membrane properties are going to tell it to fire another action potential on its own. So if we had just the cardiac myocyte, the contractile one, there would be a time period between the action potentials and the contractile relaxation events that would be unique to that particular cell. And the pacemaker signal getting to it has got to have a higher frequency than the built-in frequency. Otherwise, you're going to get the ventricular myocytes determining their own pace, and that's not good. Because what it will mean is that the ventricle will contract before the atrium has completely emptied into it, and your whole flow system, your flow circuit, is messed up temporally. So here we are following a leaky sodium, voltage-gated sodium to depolarize, generating the peak amplitude value of the action potential, which is close to the sodium equilibrium potential, excuse me, and then potassium will contribute to some of the reversal, but that's transient, and then calcium channels will open. The calcium will bring about, after its plateau, will bring about an opening of potassium channels, another set that's going to continue the repolarization phase back to the lowest level that we could consider as the resting membrane potential, the minus 90. And you certainly should recognize that the minus 90 is very close to the equilibrium potential for potassium. Well, now let's think of the refractory period in skeletal muscle relative to the contractile event, so that we can contrast it with that of the cardiac contractile myocyte. So the skeletal muscle fast twitch fiber is shown in this panel, and the refractory period is in yellow for the action potential, and it is very short compared to the amount of time required for the development of tension. So you recall that the with a single twitch of a skeletal muscle cell, similar to one contractile event in a cardiac muscle cell, one contractile event in one cardiac contractile myocyte is equivalent to one beat of the heart. Okay? You don't want it beating 3 times in the middle of a heartbeat, otherwise you're not getting a nice smooth contraction of the ventricle that's coordinated to empty it all at once. So this is what we're leading up to. So the action potential is very short relative to the contraction relaxation event of the skeletal twitch, skeletal muscle fiber twitch. So we see membrane potential on the left, and I'm having a problem with this minus 90 here. How about you? I don't remember that the resting membrane potential of the skeletal muscle fiber was minus 90. But anyway, just correct that in your mind and look at, again, as I said, at the overshoot, but consider the overall amplitude. So the peak value is not the same as saying is that the amplitude. The amplitude is the total swing, the total deflection height. The amplitude value, the peak amplitude value, is the one we've talked about, whether it is plus 20 for the skeletal muscle fiber. So look back and see if this figure is accurate. Is that overshoot height above 0 as high as it should be relative to the scale that we see here, and should this really be minus 90? Okay, certainly should be for the cardiac myocyte, and it would be a plus 20 overshoot at the peak of the action potential. So now we are seeing our time scale, as we saw here. We're 0 to 100 milliseconds. Within 10 milliseconds, we've completed the action potential even through the refractory period for the skeletal muscle fiber. The contractile event and the relaxation event extends well beyond that 10 milliseconds, but everything is done by 100 milliseconds. With the coordinated set of events, the action potential and the contraction relaxation cycle, we are out beyond 250 milliseconds for excuse me, a cardiac myocyte. Again, one of these occurs with each beat of the heart. So what you have to ask is, what is the separation between two events, as we see here, action potential, contraction, relaxation events, to generate a certain heart rate? So if you ask the question, what would be the maximum, how many 250 milliseconds can you get into a second, can you get into a minute? This is one beat, remember? So if you had one of these every single time that one could occur, how many of these are in a millisecond, how many milliseconds are in a second, how many seconds are in a minute, so how many beats per minute could you have at the maximal capability for a contractile myocyte. And I think if you go through that series of multiplications, you'll find that it's very high, so that the interpulse interval, if you want to think of it that way, for the time between action potentials in the heart is stretched out a bit at basal heart rate and is compressed a bit at higher heart rates. So what could be the maximum heart rate that you could possibly generate? You wouldn't be able to survive it, but at least mathematically, what could it be? So quite obviously, we're talking about a longer duration event of the contraction and relaxation, so you can get the emptying of the ventricle, the full contraction of the ventricle, all the cardiac myocytes contracting when they should. And I'm not saying that every one of the cardiac myocytes in the left ventricle contract at exactly the same time, but very, very close with each other. And basically it's so that you get the apex of the ventricle beginning to contract first, and then the contracted wave does, although spiraling, project back toward the base of the heart, where you have the AV valve closing and the semilunar valves opening. So keep that in mind as well. And it's when one cardiac myocyte is contracting, it's passing the action potential signal, the sinoatrial pacemaker signal, to the next cell.